A very warm welcome to all. Today I am going to give you an introduction on one of the interesting component of microbiology, that is modern classification of microorganisms. Taxonomy, that is taxis in Greek, is arrangement or order. Nomos means law, to distribute, is the science of lawful arrangement of living beings, which referred as classification of biological entities. Due to the vast diversity of living organisms, it is essential to classify or arrange them into groups based on their mutual similarities. Taxonomy consists of three separate but interrelated areas: classification, nomenclature, and identification. Taxonomy is significant because number 1, it allows one to organize large amounts of information correctly about organisms, as all the members of a particular group share many characteristics. A more accurate classification helps to gain more knowledge and usefulness of the organism. Second, one can make predictions and frame hypotheses for further research based on knowledge of similar organisms. If a relative organism has some characteristics, the microorganism in question also may have the same characteristics. Third, it places microorganisms in meaningful, useful groups with precise names so that microbiologists can work with them and communicate efficiently with other microbiologists. Fourth is, taxonomy is necessary for precise identification of microorganisms. For example, in the field of medical microbiology, where the treatment becomes exceptionally difficult when the pathogen is unknown. The term systematics often is used in place of taxonomy. In general terms, it is defined as the scientific study of organism with the ultimate object of characterizing and arranging them in an orderly manner. Knowledge acquired through study on the nature of an organism is used in taxonomy is also a part of systematics. Thus, it covers disciplines such as morphology, ecology, epidemiology, biochemistry, molecular biology, and physiology. Currently, the modern microbial taxonomy is in chaos because of the use of a new molecular techniques in classifying microorganisms. Even though these new advances have generated much excitement and are drastically changing microbial taxonomy, traditional approaches still have value. The first classification system that gained popularity before few decades is the 5-kingdom system suggested by Robert Whittaker in the 1960s. The studies of Carl Woese in 1980 and his collaborators on rRNA sequences in prokaryotic cells suggested to classify the prokaryotes into 2 distinct groups and so gave the modern classification of all the life forms, the universal phylogenetic tree of life. The universal phylogenetic tree is divided into 3 major branches representing the 3 primary groups: bacteria, archaea and eukarya. The archaea and bacteria first diverged, then the eukaryotes developed. These 3 primary groups are called domains and placed above the phylum and kingdom levels. The domains differ markedly from one another. The domain bacteria contains prokaryotic cells with bacterial rRNA and membrane lipids that are primarily diacylglycerol diesters. Prokaryotes having isoprenoid glycerol diethers or diglycerol tetraether lipids in their membranes. Archaea appear likely that modern eukaryotic cells ascended from prokaryotes. Now we will see taxonomic ranks. In preparing a classification scheme, one places the microorganism within a small homogeneous group, that itself a member of large groups in a non-overlapping hierarchical arrangement. A category in any rank combines groups in the level below is based on shared properties. In prokaryotic taxonomy, the most commonly used levels or ranks in ascending order are species, genera, families, orders, classes and phyla. Microbiologists often use informal names in place of formal hierarchical ones. Typical examples of such names are purple bacteria, spirochetes, methane oxidizing bacteria, sulfate reducing bacteria and lactic acid bacteria. The basic taxonomic group in microbial taxonomy is the species. Prokaryotic species are characterized by phenotypic and genotypic differences. A prokaryotic species is a collection of strains that share many stable properties and differ significantly from other groups of strains. A genus is a well-defined group of one or more species that is clearly separate from other genera. Microbiologists name microorganisms using the binomial system suggested by the Swedish botanist Carl Linnaeus. The Latinized, italicized name consists of two parts. The first part, which is capitalized, is the generic name and the second is the uncapitalized specific description. Example, Escherichia coli. Berge's Manual of Systematic Bacteriology contains the currently accepted system of prokaryotic taxonomy. We will see classification systems. Once data on taxonomically related characteristics of microorganisms have been collected, they can be used to construct a classification system. The most desirable classification system, called a natural classification, arranges organisms into groups whose members share many characteristics and reveals as much as possible the biological nature of organisms. Next is methods of classification. There are two general ways in which microbial classification systems can be constructed. That is, they can be clustered together. First is based on overall similarity to form a phenetic system, and second is based on probable evolutionary relationships to produce a phylogenetic system. Computers can be used to analyze data for the production of phenetic classifications. The process is called numerical taxonomy. So, let us briefly discuss phenetic and phylogenetic classifications and also numerical taxonomy. First, we will see phenetic classification. Commonly, this classification is the most natural one with the greatest informative content or predictive value. A good classification should bring order to biological diversity and may even clarify the function of a morphological structure. For example, if motility and flagella are always associated with particular microorganisms, it is rational to suppose that flagella are involved in at least some types of motility. When viewed in this way, the best natural classification system may be a phenetic system, one that groups organisms together based on the mutual similarity of their phenotypic characteristics. Although phenetic studies can reveal possible evolutionary relationships, they are not dependent on phylogenetic analysis. They compare many traits without assuming that any features are more phylogenetically important than others. That is, unweighted traits are employed in estimating general similarity. Obviously, the best phenetic classification is the one constructed by comparing as many characteristics as possible. Organisms sharing many characteristics make up a single group or taxon. Second is numerical taxonomy. Numerical taxonomy is a quantitative approach which has become possible due to the development of computers. According to Sneath and Sokal, numerical taxonomy is the grouping by numerical methods of taxonomic units into taxa on the basis of their character states. Information about the properties of organisms is converted into a form suitable for numerical analysis and then compared by means of a computer program. The resulting classification is based on general similarity as judged by comparison of many characteristics, each given equal way. This approach was not achievable before the arrival of computers because of the large number of calculations involved in forming the correct group. The process begins with a determination of the presence or absence of selected characters in the group of organisms under study. A character usually is defined as a quality about which a single statement can be made. Many characters, at least 50 and preferably several hundred, should be compared for a precise and reliable classification. It is best to include many different kinds of data such as morphological, biochemical and physiological. Organisms with more similarity are grouped together and separated from dissimilar organisms. Such group of organisms are called phenons. Numerical taxonomy has already demonstrated to be a powerful tool in the microbial taxonomy. Phylogenetic classification. The term phylogeny refers to the evolutionary development of a species, as in Greek phylon means tribe or race and genesis means generation or origin. After the book published by Darwin on the origin of a species in 1859, biologists began to develop phylogenetic or phyletic classification systems. These are systems based on evolutionary relationships rather than a general similarity. This has proven difficult for prokaryotes and other microorganisms, primarily because of the lack of good fossil record. The direct comparison of genetic material and gene products such as RNA and proteins overcomes many of these problems. Because phylogenetic results vary with the data used in analysis, many taxonomists believe that all possible valid data should be employed in determining phylogeny. In the approach called polyphasic taxonomy, taxonomic schemes are developed using a wide range of phenotypic and genotypic information ranging from molecular properties to ecological characteristics. Berge's Manual of Systematic Bacteriology in 1923, David Berge, professor of bacteriology at the University of Pennsylvania, and his colleagues published a book, Berge's Manual of Determinative Bacteriology, for classification of bacteria, that is used for identification of bacterial species. The 9th edition of this manual was published in 1993, which is still used as further edition is not published. The prime significance of the Determinative Bacteriological Manual is that it serves as a reference to aid in the identification of unknown bacteria. The first edition of Berge's Manual of Systematic Bacteriology, with more detailed work that contains description of all prokaryotic species, was published in 1984. The first edition of Berge's Manual of Systematic Bacteriology offers a primarily phenetic classification. Moreover, the classification shows many taxa that are not phylogenetically homogeneous. The classification is chiefly based on easily determined features such as cell shape, gram staining characteristics, oxygen relationships, motility, and the mode of energy production are used to classify prokaryotes. There has been enormous progress in the prokaryotic taxonomy thereafter. Especially the sequencing of rRNA, DNA and proteins has made a phylogenetic analysis of prokaryotes possible. As a consequence, the second edition of Berge's Manual of Systematic Bacteriology, which is the current modern microbial classification, is largely phylogenetic rather than phenetic and with involvement of more ecological information. The second edition has a different organization than the first edition. The second edition is published in 5 volumes. In the year 2001, volume 1 is published, which describes the archaea and deeply branching and phototrophic bacteria. Volume 2 is published in 2005, classifies the proteobacteria in 3 parts. In 2009, volume 3 was published, which detailed the firmicutes, the bacteroidetes, spirochetes, tenericutes, that is mollicutes, acidobacteria, fibrobacterus, fusobacteria, dictyoglomi, gamma timonidates, lentisferae, Verrucomicrobia, Chlamydia and Planktomycetes are included in volume 4 that was published in 2011. Volume 5 included the Actinobacteria, which is published in the year 2012. The domain Eubacteria is divided into 23 phyla. For instance, the large phylum Proteobacteria, consisting of 1500 species in 443 genera, is divided into 5 classes, namely alpha, beta, gamma, delta and epsilon. While phylum Firmicutes is divided into 3 classes, provides descriptions of more than 1300 species assigned to 153 genera. Comparisons of nucleic acid sequences, particularly 16S rRNA sequences, are the foundation of this new classification. In spite of all this development, the bacterial classification is still today in dynamic stage and as only less than 10% microorganisms is known. Today, the bacterial classification is changing as more and more organisms are cultivated and discovered. Even the name and higher taxa may also change in future.
To summarize, taxonomy is the science of biological classification, is composed of 3 parts: classification, nomenclature and identification. Living organisms are divided into 3 domains presently: the eukarya, bacteria and the archaea. A bacterial species is a collection of strains that have many stable properties in common and differ significantly from the other groups of strains. Microorganisms are named according to the binomial system. The 2 major types of natural classification are phylogenetic systems and phenetic systems. Classifications may be constructed by means of numerical taxonomy, in which the general similarity of organisms is determined using a computer. Morphological, physiological, metabolic and ecological characteristics are widely used in microbial taxonomy. Bergey's Manual of Systematic Bacteriology gives the accepted system of prokaryotic taxonomy. Comparisons of nucleic acid sequences, particularly 16S rRNA sequences, are the foundation of this new modern bacterial classification. Bacterial classification is dynamic and changing as more and more bacteria are being cultured and characterized. Thank you.